Hello everyone, welcome back to Pharma Guide. Today we are going to study about histamine. In this video we will study about the synthesis, storage, release and metabolism of histamine. So in our last video we studied about the introduction of the autocoids. We will revise quickly. What are autocoids? We discussed in our previous video that autocoids are the chemical mediators that are synthesized and function in a localized tissue or area. They work in a restricted area, or we can say that they act locally, and therefore they are also termed as local hormones. Typically autocoids are short-lived, they are rapidly degraded, and they participate in physiologic or pathophysiologic responses in the body. Today we will discuss about the first autocoid to be discovered, that is histamine. Histamine is classified in amine category of autocoids. Chemically, histamine is beta-imidazoyl ethylamine. It is found in a great variety of living organisms. It is found in animal kingdom, in many plants, bacteria, as well as in insects. English scientists George Barger and Henry H. Dale first isolated histamine from the plant fungus ergot in 1910, and in 1911 they also isolated histamine from the animal tissues. Plants that produce histamine includes stinging nettle. Histamine is present in the hair-like structure on the nettle leaves, and this is responsible for the swelling and itching produced when we come in contact with the leaves. Histamine is also irritating ingredient present in the venom of many species of wasp and bees. Synthesis of histamine. Now we will study. Almost all the mammalian tissues contain histamine. Its concentration in plasma and other body fluids is very low, but in cerebrospinal fluid its concentration is significant. Histamine is formed by decarboxylation of amino acid histidine by the enzyme L-histidine decarboxylase. Histidine is converted into histamine by decarboxylation by the enzyme histidine decarboxylase. Storage of histamine. The mast cell is the predominant storage site for the histamine in most of the tissues. In the tissues, histamine is stored in mast cell, and in the blood, histamine is stored in the basophil. Concentration is particularly high in the tissues that contain large number of mast cell. Tissues that contain large number of mast cell will have large concentration of histamine, like skin, bronchial mucosa and intestinal mucosa. It contains large concentration of histamine. In mast cell, histamine is positively charged. It is held by an acidic protein and heparin. Heparin is negatively charged within the intracellular granules. Non-mast cell histamine occurs in histaminocytes in the stomach and in histaminergic neurons in the brain. So in mast cells, histamine that is positively charged is held by an acidic protein and heparin that is negatively charged within the intracellular granules. And when the granules are excreted by exocytosis, histamine is released. Okay, now histamine release. Histamine is released from the mast cells by the process called exocytosis during the inflammation or the injury in the body, or during the allergic reactions or immune responses. It is also released by taking some of the drugs, or by the plants and the animal stings or the venoms. These are the main causes of histamine release. Now we will study in detail histamine release in the allergic reactions. Histamine is released from the cells in response to the antibodies. IgE, immunoglobulin E antibody. This antibody may be secreted in response to the invading pathogen. So the sensitized mast cell or the basophils, these are coupled with IgE antibodies. And when they are exposed to the proper antigen, when any antigen like virus, bacteria and any allergic substance like the pollen grains, it enters the body or it invades the body, it causes degranulation of the mast cells or exocytosis. By the exocytosis process, degranulation of the mast cells occurs, histamine is released. This is referred to type 1 IgE mediated hypersensitivity reaction. So when any pathogen enters the body like bacteria, virus or any allergic substances like pollen grains enters the body, our mast cell releases antibodies. Mast cell releases antibodies and these antibodies binds with the antigens to remove them. Antibodies will bind with the bacteria virus to remove them. In this process, histamine is also released from the mast cells, and this causes visual symptoms of localized allergic reactions, including runny nose, watery eyes, constriction of bronchi, and tissue swelling. We have already experienced these symptoms during any viral infections in our daily life. Histamine also contributes to generalized allergic reactions such as anaphylaxis. What is anaphylaxis? Anaphylaxis is a severe, potentially life-threatening allergic reaction. It can occur within seconds or minutes of exposure to something you are allergic to, such as any food like peanuts or any insect stings like bee stings. Anaphylaxis causes the immune system to release large quantity of histamine, which may result in blood pressure decrease in the blood pressure drops suddenly, airways become narrow, blocking the breathing. These signs and symptoms include a rapid and a weak pulse, skin rashes, nausea and vomiting. Commonly, anaphylaxis is triggered during the intake of certain foods which you are allergic to some medications, or the insect venom and latex. Okay, now we will study about the mechanism of antigen antibody reaction, how this antigen antibody reaction occurs and how the histamine is released from the mast cells and the basophil cells. Now, as a part of allergic reaction or allergic response to any antigen, when any antigen enters the body, our mast cell generates IgE antibodies. When any antigen will enter the body, our mast cell will generate IgE antibodies, and these antibodies then will bind to the surface of the mast cell with the help of a specific high protein affinity receptor, that is Fc receptor. With the help of this Fc receptor, these antibodies generated will bind to the surface of the mast cells or the basophil cells. Okay, now what happens when this antigen binds with the antibody? Activation of the signaling pathway occurs in the mast cells and the basophil cells. This involves tyrosine kinase and subsequent phosphorylation of multiple protein substrates. What happens? Activation of tyrosine protein kinase occurs, which phosphorylates and activates phospholipase C. Subsequently, phosphatidylinositol bisphosphate is hydrolyzed and IP3, that is inositol trisphosphate, is generated. This inositol trisphosphate is responsible for intracellular release of the calcium within the cells, and this calcium results in exocytosis of these granules. What is happening? Antigen will bind to the antibody, activation of signaling pathway occurs, tyrosine protein kinase activates, phosphorylates and activates phospholipase C. Subsequently, phosphatidylinositol bisphosphate is hydrolyzed and inositol trisphosphate is generated. This IP3 will cause release of the calcium within the cells, and this calcium will result in exocytosis of these granules, and release of histamine will occur. Now, how this exocytosis will occur? The calcium ions, these intracellularly released calcium ions, these will cause fusion of these granules with the plasma membrane of the mast cell, and exocytosis will occur. Exocytic release of the granule contents will occur. Now, within these granules, histamine is present in complexed form. Histamine is positively charged. It is complexed with negatively charged protein or heparin, and release of the histamine will occur when there is exchange of histamine with the extracellular sodium or calcium. Okay, cationic exchange with the extracellular sodium or the calcium sets histamine free to the target cells, free on the target cells to act. Now next is histamine release in the inflammation. Most of the effect of histamine on the blood vessels is observed in the form of inflammation or swelling. When a tissue gets injured, there is a local reaction of the tissue to the injury. Injury can be caused by any physical damage or infection or the allergic reaction, and that local reaction results in the inflammation. What happens actually? Injured tissue mast cell releases histamine, and the surrounding blood vessels, the blood vessels surrounding the damaged or the injured tissue, they dilates. Blood vessels dilates, and also permeability of the blood vessels is increased. Due to increase in the permeability of the blood vessels, leukocytes or white blood cells and some of the blood plasma proteins, they leak from the blood stream through the vessel walls, and they migrate to the site of tissue injury or the infection. Okay, so when there any injury occurs, the blood vessels around the injury dilates, and the leukocytes and the blood plasma proteins, they leak from the blood vessels to the site of the injury. That causes inflammation. Why they migrate there? To fight the infection and to nourish and heal the injured tissues. Okay, now we will see what are the major events in the local inflammatory response. So there is a minor injury caused by the skin, and the skin is infected with the pathogens. These pathogens will enter the tissue. When the pathogen enters the tissue, the chemical signals are released by the activated macrophages and the mast cells at the site of injury to the blood capillaries. So the blood capillaries will dilate, as well as the permeability of the capillaries will be increased, so that the leukocytes and the plasma proteins will move to the injured area. Clotting will occur, as well as phagocytosis of this pathogen will occur, and healing of the injured tissue will take place. So these are the major events in the local inflammatory response. These were the two main categories of the histamine release in allergic reactions, histamine release in inflammation. Now, histamine release by administration of some of the drugs, histamine release by the peptides, by the venoms and other agents. Many compounds, including large number of therapeutic agents, stimulate release of histamine from the mast cells directly and without prior sensitization. Due to administration of some drugs, histamine is released from the mast cells without sensitization. What these drugs do? Drugs usually strong bases like morphine, polymyxin, tuberculin, codeine, lidocaine, penicillin, and their vehicles are capable of releasing histamine. But this release does not involve degranulation or mast cell injury. But really what happens? These drugs displace or compete with the histamine for binding with the heparin. Histamine is stored in the granules in complex with heparin. So these drugs, they displace histamine and they complex with the heparin and get stored in the granules. So these drugs displace histamine directly without any sensitization of the mast cell. The release of the histamine from the mast cell is direct. This phenomena can result in unexpected anaphylaxis reaction. For example, vancomycin-induced Redman syndrome. Vancomycin-induced Redman syndrome. What happens in the Redman syndrome? Hypotension occurs, blood pressure falls, and flushing occurs in the upper body and the face. This is due to release of the histamine. Now, basic polypeptides are also histamine releaser. For example, bradykinin and kalatin. These are the plasma kinins. Plasma kinins are the polypeptides, and two important plasma kinins are kallidin and bradykinin. Bradykinin is a poor histamine releaser, whereas kallidin and substance P with more positively charged amino acids are more active in releasing histamine. Plant and animal stings are also capable of releasing histamine. We already know this. We already studied it in our last video. Now, histamine metabolism. Histamine is metabolized via two pathways. Predominant or the major pathway is methylation. Methylation to form N-methylhistamine by enzyme histamine N-methyl transferase. Histamine N-methyl transferase convert histamine into N-methylhistamine, and then this N-methylhistamine is converted into N-methyl imidazole acetic acid by enzyme monoamine oxidase. This is the predominant pathway. Histamine to N-methylhistamine, and from N-methylhistamine to N-methyl imidazole acetic acid. Next path, second pathway is oxidative deamination, catalyzed mainly by diamine oxidase. Oxidative deamination of histamine by diamine oxidase forms imidazole acetic acid. Then this imidazole acetic acid is converted into imidazole acetic acid riboside. This is the second pathway. These metabolites have little or no activity. These are excreted in the urine. Thank you for your time and attention, guys. If you like these videos, please subscribe to my channel, share these videos, like, and stay connected for the next one.